So for number 6, you basically have compare and contrast the C3, C4, and the CAM carbon fixation methods of photorespiration. So you can see that very clearly in figures 820 and 821. They have to be able to determine as far as exactly what's happening as far as the differences in the carbon fixation process, and they should also be able to tell the differences as far as the structure goes in each one of the leaves of these plants to allow for these processes to occur, and what environmental conditions allow one version of photorespiration versus another. Okay, C3 versus C4 versus the CAM processes. Okay, and whether it happens during the day or during the night is also very important as well. And that is also dependent upon what environmental adaptations that particular plant had, whether it's an arid environment or not, and that's an important concept and an important point to make to your students as far as the environmental adaptations that they needed to get to be able to survive in that environment, and this is one of them, the photorespiration process with the carbon fixation. Now, compare the process of photosynthesis in green plants and anoxygenic bacteria. So at the beginning of chapter 8, you have some basic descriptions as far as the processes of photosynthesis, how you do have oxygenic and anoxygenic photosynthesis. It's literally right on page 144. So now that they've gone through the process of photosynthesis, they can take a look at essentially abnormalities within the system, where you have oxygen not actually present because it's toxic to these particular bacteria. Okay, thus the reason you have anoxygenic photosynthesis in some of these bacteria. And you can go through that, it's all on page 144 and of course on the answer sheet. Now we move on to chapter 31 on fungi. Chapter 31 starts off by talking about the 5 general characteristics of all fungi, and literally bulleted 1 through 5. Okay, go through each one of the points. As far as the nuclear mitosis concept goes, you can go back and refer to figure 10.3, the fourth column or whatnot, that will show you nuclear mitosis with yeast, which is a type of fungus. You have 5 major phyla of fungi: Chytridiomycota, Basidiomycota, Ascomycota, Zygomycota, and Glomeromycota. Okay, in which each one, you have to know characteristics of each one, functions of each one, and the basic life cycles of each one. Okay. The Deuteromycetes is not an actual phylum. It's essentially a polyphyletic group, a group that doesn't have the most recent common ancestor or all of the descendants within the group. It's basically just a group of generally unrelated organisms that are placed into the group. And in this case, essentially because they're placed in this group because you don't have the description of the sexual life cycle. Okay, which is why we actually are able to classify the majority of these fungi into the different groups. Let's see. And so all 5 of the characteristics are listed on page 604 and on the answer sheet. And again, elicit as much from your students as possible. So number 9 has the structure of the mycelium. The mycelium is an accumulation of hyphae, hyphae being the basic structure of your fungi, and it's basically a chain of cells. Okay, divided by septa, which are just basic divisions that do have basic pores in between to allow for greater cell communication between the cells, which actually allows for fungi to grow as fast as they do. Let's see, and so they should be able to basically name the structure and the basic function of each one of these different parts of the mycelium. The dikaryon, or the dikaryotic stage, is the N plus N cell. So it's 2 haploid nuclei that are included in one cell. Okay, and that would be a dikaryotic cell, N plus N. then you have the typical haploid and diploid cells, haploid N, diploid 2N. Let's see, all concepts that you should review at that point. Now, describe the major life cycles. So the key is you need to know when fertilization or plasmogamy happens. You need to know when karyogamy happens, so the fusion of the haploid nuclei to make the diploid organism, and meiosis, the switch from diploid to haploid. Those are the most important parts. You need to know when these things happen, and the idea is what happens with each one. Meiosis, the switch because of the reduction division concept, the switch from diploid to haploid. The karyogamy, the switch from the dikaryon stage to the diploid stage, and the fertilization, the plasmogamy concept, where you have the two separate haploid cells fusing together to make the N plus N, the dikaryon, the dikaryotic cell. Okay. Not only that, but they should know as far as structures go. So these different phyla are basically named after each one of their basic sexual units. So the Basidiomycota, and after the Basidium, the basic sexual unit of the Basidiomycetes. The ascus is the basic sexual unit of the Ascomycetes, etc. So that's something that they should basically know. Now, inquiry the inquiry question in number 11 is follow the graph. It's really easy to interpret the graph. You shouldn't have any problems just as long as you're able to read the graph. 12, the basic symbiosis concept. Again, as much as you can from the students, asking what symbiosis is, the differences between mutualism, commensalism, and parasitism, and focus in on the good ones, the mutualisms, the lichens and the mycorrhizae. Okay, and I'll keep it at that for this video, and we'll finish up the workshop in the next video class.